In Part 4, we will use the Gene Record Finder, which is a web tool that enables us to quickly identify the set of exons for a given gene and to retrieve their coding DNA sequences, or CDSs, also referred to as coding exons. The Gene Record Finder will also provide details such as number of isoforms, exon-intron structure and their coordinates, and transcript and protein information of the gene in question, in this case Reb. It is important to remember that the details provided by the Gene Record Finder are for the gene in the reference species, D. melanogaster. We will use the details from Reb in D. melanogaster to assist us with creating a gene model for Reb in D. jacoba. Before we can construct an orthologous gene model, we will need to ascertain the gene structure, the number of isoforms and CDSs of the D. melanogaster Reb gene using the Gene Record Finder. Open a new web browser tab and navigate to the Gene Record Finder. Enter Reb into the text box. Click on the Find Record button. The Gene Record Finder shows that Reb has two isoforms, A and B, in D. melanogaster. A graphical overview of the two isoforms is shown in the mRNA Details panel. The CDS usage map, under the Polypeptide Details tab, shows that both isoforms have the same set of coding exons. The coding exons are ordered from 5' to 3' from left to right in the CDS usage map. Hence, the differences between these two isoforms are limited to the untranslated regions. Based on parsimony, minimizing the number of changes compared to D. melanogaster, we expect to find both the A and B isoforms of Reb in our D. jacoba genome sequence. For this walkthrough, we will only focus on annotating the CDSs, which do not include the UTRs. Consequently, we only need to determine the coordinates of the 5 CDSs for one of the isoforms, because the set of coding exons for both the A and B isoforms are identical. FYI, if the coding sequences of all isoforms aren't identical, you'll need to annotate each unique isoform. Uniqueness is based on the coding sequence.